Welcome back. We are now starting with session 2. Our first speaker is Joaquín Vierna. Thank you. So Joaquín did his PhD in DNA barcoding, population genetics, and evolutionary genetics of NC's razor shells. In 2011, he co-founded AllGenetics and has been AllGenetics general manager since then. During this year, together with his colleagues at AllGenetics, Joaquín has specialized in high-throughput sequencing and bioinformatics, and has carried out dozens of DNA metabarcoding projects for both research teams and the industry. His talk is titled Index Swapping in DNA Metabarcoding. Thank you.
Okay, so hello. Yeah. In this talk, I will discuss index swapping in the context of Illumina DNA metabarcoding analysis. First, I will give a brief introduction to this topic, and then I will present the results of some recent experiments that we have carried out at AllGenetics in order to better understand index swapping. So back in 2015, we were asked to analyze over 4,000 mite samples through DNA metabarcoding. But when we analyzed the first high-throughput sequencing results, we were really stressed as we found apparently extensive cross-contamination between the samples. So we decided to increase our anti-contamination measures in order to make sure that this kind of contamination was not coming from our own wet lab, and then we analyzed the same samples again. But the contamination was still there. However, we soon realized that cross-contaminations always happened between samples which had been sequenced together in the same MiSeq run. Okay, cross-contaminations always happened between samples which had been sequenced together. So we wanted to understand what was going on there. We screened the literature, and we found some recent papers at that time where the authors discussed a phenomenon known as index swapping, but also mistagging or index switching, index crosstalk, tag jumping. They're all different names for the same phenomenon, by which a small portion of reads is assigned to the wrong sample during high-throughput sequencing. Okay? A small portion of reads is assigned to the wrong sample during sequencing. So index swapping has been observed to occur at a rate of 0.2 to 6%. On the slide, you see the typical results of a DNA metabarcoding analysis. Here you have two samples, sample 1 and sample 2. Sample 1 has two different species, species A and species B, and each of the two species account for 50% of the sequencing reads. And then you have sample 2 with one species only. On the right-hand side of the slide, you have the same analysis, but in this case with a 5% rate of index swapping. So now sample 2 has two more samples. However, these samples are artifacts. They are not real, and they were, of course, not occurring in the original DNA sample. So, as you can imagine, index swapping may be a problem in DNA metabarcoding studies, as a species might be detected in samples in which they did not occur. But fortunately, there are bioinformatic filters which reduce or mitigate the effect of index swapping. So the mechanisms by which index swapping occurs are not 100% clear, okay? But some authors mention the following. Index cross-contamination, either during oligonucleotide synthesis or during handling at the time of library preparation. Sequencing clusters derived from more than one molecule. Chimera formation during library preparation, run-to-run carryover, errors during index sequencing, and then in the particular case of HiSeq 4000, HiSeq X, and NovaSeq sequencers, which have patterned flow cells with exclusion amplification chemistry, the chimera rate is increased, and so is the rate of index swapping. So now I'd like to discuss three other factors which, according to the literature, they do not cause index swapping, but they make it worse. We have called them potential aggravating factors. So the first one is single indexing during library preparation as opposed to dual indexing. So here you have two libraries. The first library, we call it a single index library because it has only one index, which is the forward index in this case. And then you have here a dual index library with two indices, the forward and the reverse indices. We would expect more index swapping if we are working with single index libraries. The second potential aggravating factor is dual indexing with saturated index combinations as opposed to dual indexing with non-saturated index combinations. Here we have three libraries, and we say they are saturated because they have the same forward index and three different reverse indices. However, these other three libraries are non-saturated because they have three different forward indices and also three different reverse indices. And in this case, we would expect more index swapping if we are working with saturated libraries. And the third potential aggravating factor is non-equimolar library pooling in the sequencing run as opposed to equimolar pooling. Again, three libraries here, the green, the red, and the yellow library, which are combined in a sequencing pool. However, as you can see, this pool is non-equimolar because not all the three libraries contributed with the same amount of DNA to the pool. The green library contributed with more DNA. This other pool is an equimolar pool because the three libraries contributed with the very same amount of DNA. Okay? And we would expect more index swapping in the case of non-equimolar pooling. So, at AllGenetics, we designed 4 different experiments to specifically test 2 potential aggravating factors, which are index saturation and non-equimolar pooling, in an Illumina MiSeq amplicon sequencing pipeline. Even though I am in charge of presenting these results today, actually my colleagues did most of the work. So Anton did the lab work, and both Anton and Neus did the bioinformatic analysis, and Jorge did the statistical analysis. We extracted DNA from 22 different fish species. And we analyzed a fragment of the mitochondrial gene 12S rRNA. We selected this gene, or this fragment, because the primers worked very well in all the 22 different species, but the region was still variable enough to differentiate between the 22 fish species. So basically, this is what we wanted to look at. For example, in the case of the eel library, we wanted to see if we get only eel sequencing reads, or we get many eel sequencing reads, plus some reads from other species which were sequenced in the same pool. This is the experimental workflow. We have a step of DNA isolation from each of the 22 fish samples, then the preparation of amplicon libraries, and here we followed two different approaches. A non-saturated approach with 25 different forward indices and 25 different reverse indices, and a saturated approach with one forward index only and 25 different reverse indices. I am saying 25 instead of 22 because we had 3 negative controls or blanks. And then the libraries were quantified and pooled. Again, 2 different approaches, equimolar pooling and non-equimolar pooling. In the case of non-equimolar pooling, the library which contributed with more DNA to the pool was 30 times more concentrated than the library which contributed with less DNA to the pool. And then the different pools were sequenced in the Illumina MiSeq sequencing platform. So we actually have 4 different experiments, but today I will present the results of the first 3 experiments because the results of experiment number 4 are not available yet. So this is what we observed. We observed a maximum index swapping rate of 1.6, one of the libraries, a minimum rate of 0% because several libraries didn't show any index swapping at all. And then this is the average value and the standard deviation. I just wanted to say that index swapping can be handled by eliminating OTUs occurring at low frequencies in each sample. So, for example, you might want to get rid of everything at a occurring at a frequency below 1.6 in order to be on the safe side. Here we have the reads incorrectly assigned to each of the 22 libraries in each of the 3 experiments. We didn't observe differences between the 3 experiments, even though we were actually expecting some differences. But if we look in detail into the results of experiment number 3, we see this correlation. These are the reads swapped to other libraries and the DNA amount of each library in the pool. So the more concentrated libraries in the pool swapped more reads to the other libraries. That's why it is important equimolar pooling. And here we have the reads incorrectly assigned, and we do a comparison between the 3 experiments. And I just wanted to show you this correlation here. In order to better explain this, I'm showing this table just for one sample, okay? The thing here is that experiment 1 and experiment 3 were made with the same libraries, but in the case of experiment 3, they were pooled in non-equimolar amounts. So, but they are the same, very same libraries. So I show you the results for sample 1 only. Sample 1 belongs to this genera. As you can see, we get a lot of reads from assigned to this genus, here. In both experiment 1 and experiment 3. But then we also get some reads incorrectly assigned to the other libraries, which are highlighted in green or yellow. And there is a strong correlation between this index swapping, as you can see. So the index swapping pattern seems to be consistent between these experiments, okay? So to conclude, according to these results, we have not observed differences in the rate of index swapping between saturated and non-saturated libraries, which contrasts with some recent papers, which suggest that non-saturated index combinations are much better. The libraries which are more concentrated in the pool tend to swap more reads to other libraries, so my recommendation would be to do always equimolar pooling. And then index swapping is consistent between pools prepared from the same libraries but sequenced in different MySeq runs. So it seems that it is not a random phenomenon in the sequencer. And well, we still have to analyze the results from experiment number 4 in order to better understand what is the main mechanism causing this index swapping in our experiment. Thanks.
Thank you, Joaquín. Are there any questions? Yeah?
Yes.
You mentioned this random carryover, so how big problem is that?
I'm not sure how big it is, but it's, I mean, this is factors that we, that there are in the papers, authors suggesting what could be causing index swapping, and it's just contamination in the sequencer machine. Not in the flow cell because you don't reuse it, but in other parts of but I'm not sure how important it is actually.
But I have seen quite high contamination in my samples, and that was the principal cause I suspected to be.
And how about, for example, contamination during index synthesis in the company? I mean,
it could be possible, of course, in any of the steps. Yes.
For example, we tried to contact our supplier to really understand how they synthesize the indices, which are primers actually, and they didn't give a lot of explanations. So,
Probably it's a very silly question, but do you think it has something to do with the instrument? I mean, you talk about MySeq, but comparing this MySeq with that other MySeq in a different lab, do you think it?
Anton?
You have that information actually. These samples might have been run in different machines actually.
And you don't
but we
think that's a
don't know.
factor? So you don't think that's a factor?
They were running different machines. Well, I guess if we're seeing such a strong correlation between between experiment 1 and experiment 3, and we think they were running different machines, we don't think the actual machine is a factor. Okay. What it may be a factor is the model of the sequencer. So the rate of index swapping is going to be greater in the HiSeq 4000 and NovaSeq because they use a different chemistry. And that's actually known, and Illumina has released a paper about it. But yeah, that might be a factor of these.
Over there?
Yeah, well, I was going to ask more or less the same. So if swapping is not random and it cannot be attributed to the specific machine that you're using,
I don't know. I mean, according to these results, but they're still preliminary, it seems it is not random. But
would it be something related to the specific taxonomy, like that genius that it's...
I think it might be related, for example, to the index sequences, that some, perhaps there are some consistent errors in the sequencer, you know?
You change the tags.
Yeah, or contamination, or some small degree of contamination in the lab or in the company where they synthesize the indices.
You would expect them being random, right?
Yeah, that is what I was expecting.
And what about the sequence of the amplico? Do you think that this has an influence on the...
Well, I think the insert could always have an influence if it tends to form chimeras, for example. But yeah, it could be. I mean, the thing is that it seems it could be several things, perhaps happening at the same time.
Is this the same as mistagging, or?
Yeah, it's the same thing. Yeah. Okay. Yeah, sorry. Because at AllGenetics we used to call it mistagging, but then I read papers using index swapping, so that's why I was referring all the time to index swapping.
So, do you have a preferred method of finding what threshold of OTUs you can throw out without doing an entire experiment for each project? Yeah,
I mean, I think it would be great if you could run like a mock community, or in each experiment, so you specifically test this every time. For example, in our case, in this particular case, the blanks didn't have any read. So
the other thing we've thought about is to add to the sequencing sample sheet. So when you specify which indexes you've used, to add some indexes that you haven't actually used and see if those get any reads. We've been told by Illumina not to do that. Apparently the software tries to find the best match within the indexes you've specified or something like that. I don't understand it. But yeah, Illumina advises against that. But
so sometimes with misread indexes, it can still assign it to a sample because it's the closest match, is what they sort of say.
Yeah. So yeah, a misread index might get assigned to a sample that you don't actually have in your set. So.
I forgot to say, by the way, that index swapping is worse in amplicon sequencing or DNA metabarcoding compared to genome sequencing, for example. Because if you sequence a genome, the swapped reads would be like kind of randomly distributed along the assembly, and you won't notice them actually. But in this case, it is worse.
You have mentioned to use a mock community because this is library indexes, not sample, right? So you should use a mock community that doesn't have anything to do with your real.
Yeah, or actually perhaps not a mock community, but just one, one species only in each library, as we did here.
No more questions? Okay, thank you, Joaquín.